I'm Laura, and in this video I'm going to present an article that is called A CRISPR-based approach using the Cas9 sgRNA to detect SARS-CoV-2. But first, what is CRISPR-Cas9? Clustered regularly interspaced short palindromic repeats associated to a protein 9 or CRISPR-Cas9 is a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. But how does it work? The CRISPR-Cas9 system consists in two key molecules that introduce a change mutation in the DNA. These are an enzyme called Cas9. This acts as a pair of molecular scissors and can cut the two strands of DNA at a specific location in the genome so that bits of DNA can be added or removed. A piece of RNA called guide RNA or gRNA. This consists of a small piece of pre-designed RNA sequence about 20 bases long located within a longer RNA scaffold. The scaffold parts binds DNA and the pre-designed sequences guides Cas9 to the right part of the genome. This makes sure that the Cas9 enzyme cuts at the right point in the genome. And what is SARS-CoV-2? Severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 is a highly transmissible and pathogenic coronavirus that emerged in late 2019 and has caused a pandemic of acute respiratory disease named coronavirus disease 2019 or COVID-19, which threatens humans' health and public safety. The general objective in this article is to describe a new way of using CRISPR-Cas system as a rapid and highly specific tool to detect SARS-CoV-2 virus using the in vitro Cas9 sgRNA-based technique. In the figure, we can see a comparison between PC-Press and another CRISPR-based techniques used to detect SARS-CoV-2. PC-Press has the advantage that includes an additional specific step, that is, the RE cleavage seeds are blocked, as compared to the other CRISPR-Cas-based diagnostic platforms such as Cas12 and Cas13 designed to detect the E and N gene and the S gene of the SARS-CoV-2. Methods. First, we have the cleavage assay. The protocol for in vitro cleavage consists of Cas9 with guide RNA, gRNA, ribonucleotide protein, RNP formation, followed by incubation with the target DNA. For EMSA, the reaction is directly loaded onto agarose gel for visualization of the target DNA band and the shift due to binding by the Cas9 RNP. To assay for cleavage, protein K is added to degrade the RNP and load the target DNA cleavage R or uncleaved, to migrate consistently with its molecular weight. Heating at 95 Celsius degrees rapidly inactivates the RNP on demand, allowing time-resolved measurements of Cas9 cleavage kinetics. This protocol facilitates the characterization of the like activation mechanism of photocage PF CRISPR-G RNA. Understanding the fundamental principles of Cas9 binding and cleavage behavior is essential before the widespread use of this system and can be readily accomplished in vitro through both cleavage and electrophoretic mobility shift assays. The PCR cloning. In its simplest form, PCR-based cloning is about making a copy of a piece of DNA and at the same time adding a restriction sites of the ends of that piece of DNA, so it can be easily cloned at plasmid of interest. Typically, a PCR reaction is performed to amplify the sequence of interest and then is joined to a vector via a blunt of single base overhang ligation prior to transformation. Next, we have cell culture media and DNA transfections. Transfections is the introduction of the DNA into our eukaryotic cells and is used in search, study, and modulate gene expression. The transfection techniques serve as an analytic tool and that facilitates the characterization of genetic functions, protein synthesis, cell growth, and development in a cellular environment. RT-RPA, or recombinase polymerase amplifications, is a highly sensitive and selective isothermal amplification technique operating at 37 to 42 Celsius degrees with minimal sample preparation and capable of amplifying as low as 1 to 10 DNA target copies in less than 20 minutes. This method eliminates the need for a thermal cycler and has high deployment potential to resource-limited settings. In the results, we are going to analyze some Western blots. In the figure 1, we see that increased concentrations of Cas9 sgRNA complexes block the activities of BBSL and XBEL restriction enzymes. So, figure A shows fragment M1 of the M gene. In the first column, we have PCR fragments of the M gene with known sizes. In the second column, we have PCR fragments of the M gene without the BBSL restriction enzyme, so we can see the expression of the M gene. In the third column, we have PCR fragment of the M gene with the BBSL restriction enzyme, so we cannot see the expression of the M gene. The next columns have higher concentrations of Cas9 sgRNA targeting the BBSL restriction site, so we can see that expression of the M gene increase. In figure B, we see the same process, but with increasing concentrations of the Cas9 sgRNA targeting the XBAL restriction site.
Finally, in figure C, we have 200 nanograms of PCR fragment of the full-length M-gen, subject to single or dual restriction enzyme digestions, BBSL or XBAL without or with pre-incubation, with increasing concentrations of dCas9 sgRNA BBSL or dCas9 sgRNA XBAL, or multiplexing of both.
Figure 2. We notice that dCas9 sgRNase complexes rapidly protect the M-gen from cleavage by the restriction enzymes.
The entire M-gen, 75 nanograms, was pre-incubated for A, 5 minutes, B, 15 minutes, and C, 30 minutes, with increasing concentration of multiplexing, dCas9 sgRNA BBSL and dCas9 sgRNA XBAL, followed by restriction enzyme cleavage for 1 hour at 37 Celsius degrees. This press specifically detects the M-gen expressed in HAK293 cells and from individuals test positive by RT-qPCR. A total of 75 nanograms of M2 fragment derived from something different in each case were subject to single XBAL restriction enzyme digestion without or with pre-incubation with increasing concentrations of dCas9 sgRNA targeting XBAL restriction site.
In this article, there is not an actual discussion from the authors, because in the whole article, they use the protocols previously described by the main author. However, in this table, we can see some of the protocols that they used, and you can notice how they always agreed to use them. Now, we are going to talk about my conclusions about this article. First one is that compared to RT-qPCR, this DC-press approach has the advantage that it's faster and more accurate. This is important because more time and investigation it can be used to detect viral infectious diseases and pathogens as well as SARS-CoV-2. So far, there are limitations of using the DC-press approach for molecular diagnostics to detect viruses and pathogens such as fungi. The second one is that the CRISPR-Cas9 is currently the simplest, most versatile and precise method of genetic manipulation, and for that reason, it's necessary to improve this technique and do more tests to discover further applications based on this technique. And that was the whole article. I hope you enjoyed it and understood the importance of molecular biology in the current medicine. Bye!